Hello dear professor and classmates. Today I would like to share some part of my work under the title of Role of Grassy Tiller 1 GT1 in Regulating Tillering in Rice. I'm Muhammad Rosyadi Adnan from Plant Developmental Genetics Lab under the supervisor of Professor Chang Dok Han. The first part of my presentation is the introduction. Rice is one of the most important crops that globally consumed by many people all around the world. Similar with maize, wheat and some other crops. Therefore, because the high consumption of this plant's product, the demand for the rice is continuously increasing over the recent years. And as we know, humans already start domestication of rice for a long time ago. Since the introduction of Green Revolution, various ways has been applied to tackle many problems for improving the agricultural yield of rice. Impact of Green Revolution on rice. Beginning around 1960s, Norman Borlaug proposed a concept called as Green Revolution. Green Revolution is a concept by which agricultural revolution has been done by improving many aspects such as the use of irrigation properly, the use of fertilizer for boosting the growth of the plant, and the use of insecticides and pesticides which prevent the attack of the insect and pest, and also the use of technologies, and one of the important key is the use of high-yielding varieties. High-yielding varieties is characterized by not only increased yield itself, but it has to be disease resistant, tolerant to the stress, and then pest and insect resistance, having a high grain quality in terms of rice, and then have nutrient use efficiency. Having nutrient use efficiency means that the plant can utilize maximally for any nutrient that given by the farmers, so it can grow well even with the less need of the fertilizer. And finally, the main character of high yielding varieties is increased yield. How the scientist improves the rice yield? The improvement of rice yields mainly characterized by the increase of number of panicle, number of grain per panicle, and grain weight. The formation of grain yield is mainly dependent on the genes that control tillering and panicle development. There are numerous genes related with this property such as GN1A and LOC and NOC1 that related with number of grains per panicle. There are also several QTL that is related to grain size such as GS3 and GW2, GW5 that influence grain width and length. And also we have GS2, GL2 that regulates grain size, and GIF1 and DST that increase grain filling. And another important parts of improving rice yield is the genes that are related to tillering development and plant architecture. In this case, Moch1 is a first transcription factor that is known to regulate the tiller outgrowth, and then followed by strigolactone synthesis enzyme like D3, D10 and D27 that regulates the tiller number. Also OSTB1 here derivated from maize TB1 regulates the tiller number, and also numerous genes like LASY1, LPA1 and PROC1 also regulates the tiller angle. Altogether, these properties would contribute to the improves of rice yield. Tiller and plant architecture. Initially, domestication starts with naturally occurring prostate and erect phenotype. The prostate phenotype offers space occupancies, tolerance of trampling, and weed repression. However, later, the erect phenotype is much more preferred by the farmer because it can maximize the farmland use, means that we can grow more plants within certain range of field with the plant that showing erect phenotype rather than prostate phenotype. During the introduction of Green Revolution concept, the innovations of semi-dwarf wheat and rice cultivars has improved crop production immensely since 1960s. Then, the semi-dwarf architecture does not only offer the ease of harvesting but also increase the tiller number in certain extent, which lead to the increasing of the yield. So why the crop plan architecture is important? Crop plan architecture determines planting density in the field and thus influences to a large degree the light harvest, disease resistance, and lodging tolerance. And in case of ideal plant architecture that has been proposed by many scientists, it should have less number of non-reproductive tiller, increased grain per panicle, dark green colored, thick stem, erect leaves, and vigorous and deep root system. Tillering and yield. Plant need to balance between the producing tiller and producing the seed. So they have to maintain whether the vegetative growth and reproductive growth are both balanced. And in grasses, what is called as tiller are side branches of the culm that grow at the ground level from nodes of non-elongated internodes, that determining the overall shoot architecture of the plant and affecting important agronomical features like competition with weeds, herbicide treatment, spacing, and the ease of harvesting. As an organism, plants need to balance between production of the tiller and production of the seed. Too many non-reproductive tiller are unwanted since it will absorb the nutrient allocation that may be used for producing seeds. Ideally, high yield will be achieved when the plant has more tiller number and hence has more panicle number as well as the grain per panicle and grain size. However, as an organism, plant try to balance that when plant has less tiller, each panicle would tend to produce more grain per panicle, and on the opposite way, when plant has more tiller, usually the number of grain per panicle will be reduced. So this is essential properties in tillering, given that yield is mainly controlled by number of panicle per plant, grain per panicle, and grain size itself. Here, in this research published by Cell Research in 2017, we can see that IR66 has less tiller, and then Chunjiang 06 on the other hand has more tiller number. And then they cross these varieties, and we can see from the figure P that IR66 naturally have more increase of the number of grains per panicle. On the contrast, the Chunjiang 06 have less grain number per panicle, and the real 52 or recombinant inbred line, the crossing between IR66 and Chunjiang 06 have a high number of grain per panicle similar with IR66, but the phenotypes looks like Chunjiang 06. If we notice here, RIL52 has both advantages from IR66 and Chunjiang 06. The ER66 gives the properties of number of grains per panicle that high with less tiller number per plant. However, in terms of a plant architecture, we can notice that RIL 52 is mostly similar with Chunjiang 06 in terms of erect, so it can maximize the farmland use. So, as this has been proposed earlier, the ideal plant architecture would be expected to having less numbers of non-reproductive tiller but still maintain the grain per panicle number. There are several factors determining the tiller development, which fall into three parts: the genetics, hormones, and environmental. From the terms of genetics, we have several parts of important genes in maintaining the axillary meristem, since axillary meristem establishment and maintenance are essential in initiating bud growth and bud outgrowth. And also we have several hormone biosynthesis and signaling genes that are regulating how the axillary bud would develop. On the other hand, the microRNA, several microRNAs such as microRNA 156, protein transporter, and transcription factors are involved in the tiller development. Several hormones known to involve in tiller development are auxin, cytokinin, gibberellin, strigolactone, and brassinosteroid. Auxin has been known as a hormone that is polarly transported from the apical meristem to the basipetal region. So it has a role in inhibiting or suppressing the bud outgrowth, which known as the apical dominance. The genes that are involving in auxin biosynthesis or auxin transportation would lead to the suppression of bud outgrowth, and any mutation involving in auxin biosynthesis or transportation usually lead to the growth of bud, axillary bud, more than normal. Next is cytokinin. Cytokinin, on the other hand, in contrast with auxin, it has a role in promoting axillary bud activation, which trigger the outgrowth of axillary bud. And the next is gibberellin. Gibberellic acid negatively regulated tillering, given that the high Ga are mostly associated with less number of tillering, and the lower Ga content usually associated with the higher tillering number. And also strigolactone, the next hormone, acts as hormone that inhibits side branching. The high level of strigolactone can inhibit the side branching. However, several mutations of genes involving in SL biosynthesis, known as to increasing the axillary bud, which lead to the increasing of the tiller number. And also the last hormone involved in tiller development are BR, brassinosteroid, along with the GA, controlling plant height, and mutant plant in BR synthesis exhibit dwarf phenotype. In terms of environmental factors that determining the tiller development, there are nutrients use efficiency, light intensity, and water use efficiency that are strongly affected tiller development. In terms of light intensity, here we have special focus because some of the character of the tiller development as a shade avoidance mechanism, that the plants would grow less tiller number when it grown on the darker condition, because it more focusing on the upward growth in order to gaining more light instead of growing surrounding them to expand their tiller number. So here is one of our references. Grassy tillers 1 promotes apical dominance that is firstly discovered in maize, and it relates to tiller number and response to shade signal in the grasses. As we can see here, the mutant plant in the figure C have more tiller number in comparison to its wild type version of GT1 in figure D. If we notice the number of tiller also, the tiller number of GT1 mutant in the figure in the graph G we can notice that GT1 tiller number is more than twice in comparison to the wild type, and also it affecting the shank length also. The shank length of the GT1 mutant have grown more than its wild type. So altogether, this paper suggested that Gracie tillers 1, in addition to respond in the tillering number, also regulating various biological processes such as the shade avoidance mechanism. Using the alignment of protein amino acid sequences, we found that many amino acid sequences that are conserved in both OSGT1 and CMYGT1. Therefore, we think about the possibilities of the OSGT1 that act in the same way, such as CMYGT1, and maybe have resembled the same or similar role in the shade avoidance mechanism. So next, in our lab, we generate the overexpression line and RNAi lines, and in the two weeks old plants, we can see that the differences of overexpression plants in comparison to the wild type. Overexpression plants here show less tiller number during the 2 weeks, and on the other hand, the RNAi lines during 2 weeks already shown for growing more tiller number in comparison to the wild type. So from these things, we think the similar way with the publication in maize that GT1 may act as negative regulator of bud outgrowth. Here are the constructs that we used for generating overexpression lines and RNAi lines. In the left side is rice GT1 cDNA construction that is used for generating vector for overexpression line, and in the right side we have vector that is used for generating GT1 RNAi lines. In previous published paper, they mentioned that the expression of TB1 does not change in the GT1 mutant or wild type, and on the other hand, GT1 expression is suppressed strongly in the TB1 mutant in comparison to the TB1 wild type. It suggests that TB1 acts upstream of the GT1, and in this paper they propose a model that GT1, a negative regulator of bud outgrowth, works downstream of the TB1, which known to be also a negative regulator of bud outgrowth, and upstream of those genes is phytochrome B that is activated with the high number of red-far-red light ratio, and it is inhibited in low red and far-red ratio. Using this model, we propose that maybe the Oryza sativa GT1 may act the same way as the GT1 in maize 2. So we begin for testing the relationship between phytochrome B and GT1. In addition to GT1 RNAi lines and overexpression lines, we have also phytochrome B mutant plants. So, based on the previously published data that phytochrome B related with GT1, here we do crossing between GT1 RNAi lines and phytochrome B mutant. And here we generate 4 kinds of combination, which are wild type, homo wild type, homo, and double homo. And we found that this combination of genotypes significantly causing a difference in tiller number, but not that much different in terms of plant height. Here we found that homo wild type, GT1 RNAi lines, and phytochrome B confer more tillering as a result, similar with GT1 RNAi lines single mutants. And on the other hand, GT1 RNAi lines phytochrome B in the combination of wild type and homo, however, have very suppressed
tiller number in comparison to the wild type. It means that phytochrome P supposed to be upstream of GT1. However, if we have a look on GT1 RNAi lines and phytochrome P double homo mutant, we found that the phenotype of GT1 RNAi lines wild type and phytochrome B homo supposed to be slightly recovered. It means that maybe another genes are involved in maintaining the tiller phenotype.
Based on what we have found previously on the phenotype, in which the phenotype of GT1 RNAi lines and expression lines are different, we then try to investigate the differentially expressed genes in both overexpression and RNAi lines. According to the RNA-seq data, we found that 2 genes are commonly shared in upregulated overexpression lines and downregulated in RNAi lines, and 9 genes are common in downregulated OX line and upregulated in RNAi lines. Some genes then are specifically upregulated in overexpression lines, downregulated in overexpression lines, upregulated in RNAi lines, and downregulated in RNAi lines. Some genes subsequently are selected to check its expression level in overexpression lines and RNAi lines to confirm whether these genes are related to tillering. Among the selected genes, we try to find the genes that are having the relationship with cell cycle and development, since the relationship between tillering and cell cycle are still less understood. Also, we try to find the genes that having GT1 binding motifs as a target genes of GT1 protein. We found that these 2 putative genes are differentially expressed among overexpression lines and RNAi lines. These genes, therefore, we check the expression in the wild type GT1 overexpression lines, GT1 RNAi lines, and FC1 mutants. Here, we found that these genes are downregulated in both GT1 RNAi lines and FC1 mutants. In addition to previous finding, we try to find common genes that are differently expressed in both overexpression lines and RNAi lines. And here, we found that 2 locus of putative genes that are highly expressed in GT1 overexpression lines and suppressed in GT1 RNAi lines. Similar with previous findings, these genes having GT1 binding motifs, which indicate that these genes might be the target of GT1. Using all those genes, we can study how GT1 interacts with those target genes. In previously published data, we know that ATB1 acts upstream of the GT1 because GT1 expression is suppressed in TB1 mutant. However, the TB1 expression doesn't change in comparison with between GT1 mutant and wild type. So, based on that publication, we found that the maize TB1 have ortholog in rice, which is OSFC1 or phytochrome-1. Therefore, here we want to know whether these 2 transcription factors bind to each other's promoter region. Currently, in order to find whether GT1 protein can bind to FC1 promoter region and vice versa, we will test the protein and DNA interaction using a technique called as EMSA. EMSA stands for electrophoretic mobility shift assay, which is based on the interaction of protein and DNA. The interaction of protein and DNA sequence will make the probe labeled DNA will migrate slower than it would. So, by detecting the change in migration rate, we can indicate that there is interaction between protein and specific DNA sequence. Here are the constructs that we used for electrophoretic mobility shift assay. In the left side are the construct of GT1 in the background of PGX40-1. It consists of three parts, which is N-terminal, HD-zip, DNA binding domain, and CTR. Here, we use this construct to test whether any parts of these protein regions can bind to FC1 promoter region or GT1 putative target genes.
Whereas in the right side, we have the construct of FC1 in the background of PGX40-1 vector. Here also FC1 has three parts, which are NTR, and then TCP2, DNA binding domain, and CTR. Here we will test whether the FC1 proteins can bind to the promoter regions of GT1. Currently we are still in progress for doing this experiment. Hopefully, if we get succeed in this experiment, we will know the interaction between GT1 and TB1. So hopefully we can know how both genes that are related to tillering having interaction and in response to tiller and development. Discovering the target genes of GT1 would reveal what candidate genes that are act downstream of GT1, and it will help us to further understanding how GT1 regulating tiller development or bud outgrowth in rice. The second conclusion is the study of the interaction between two transcription factors, OSGT1 and OSFC1, would reveal how those genes having possible shared target genes which responds to the tiller development. We've got to the end of this presentation, and I would like to say thank you very much for the attention.